What if the key to understanding brain diseases like dementia has actually been hiding right under our noses, deep within our brain's own chemistry?
It's a fascinating thought, isn't it?
Yeah, like could something as basic as how our brain handles sugar, its metabolism, really hold the answers for protecting it?
Well, that's exactly what we're diving into.
Today, we're taking a deep dive into this, pretty groundbreaking study. It's from the Buck Institute for Research on Aging, just published this June in Nature Metabolism. And it's really making people rethink the role of something we didn't pay much attention to in neurons before: glycogen.
That's stored sugar, yeah.
So our mission here is to unpack what they found. These surprising discoveries about how tau proteins, the ones linked to Alzheimer's and other diseases, how they interact with these sugar reserves.
Right.
We want to explore what this might mean for maybe future treatments. By the end, you'll get this cutting-edge research and hopefully a new perspective on the brain's own defenses.
And it really hints at something profound, how adaptable our brain is under stress. And, just how critical that metabolic balance is for keeping it healthy.
Okay, so let's unpack this. Traditionally, glycogen, that's stored glucose, right?
Yep, basically sugar reserves.
It was thought to have, a very limited role in neurons themselves, more for other brain cells, like astrocytes. Why did we think that, and how is this new work flipping that idea?
Well, the old view is that neurons are so energy hungry, they mostly rely on a constant glucose supply directly from the blood. They weren't thought to bother much with storing it as glycogen.
So they didn't need a backup stash.
That was the assumption. But this research suggests maybe they do. That maybe neurons do activate this reserve system, this sort of backup metabolic route, especially when things go wrong, like in these tau diseases, these tauopathies.
So it's like a hidden defense mechanism.
Kind of, yeah. It points to this idea of metabolic flexibility in neurons that we perhaps underestimated. They might be more adaptable metabolically than we thought.
Okay, but here's where it gets really interesting, right? The study looked closely at what tau was actually doing.
Exactly. That's the core finding.
They saw this in, a couple of different models: fruit flies, Drosophila, and also human neurons derived from stem cells.
Yes. Using both models adds a lot of weight to the findings. They observed that these tau proteins, when they're pathological, actually bind to glycogen. They stick to it.
They trap it.
They trap it. And the consequence of that is pretty serious. It stops the glycogen from being broken down normally.
Okay, so the fuel reserve is there, but it's locked away.
Precisely. And that jams up a really crucial pathway called the pentose phosphate pathway, the PPP.
PPP. Why is that one so important?
Well, the PPP is absolutely vital for the brain's antioxidant defenses. It generates a molecule called NADPH.
NADPH.
And NADPH is essential fuel for regenerating glutathione, which is probably the brain's most important antioxidant. It mops up damaging molecules, reactive oxygen species.
So the PPP makes the fuel.
That recharges the brain's shield against oxidative stress.
Okay, so let me get this straight. Tau acts like a sticky trap for glycogen.
Right.
Which means the glycogen can't be used by the PPP pathway.
Correct.
Which means the brain can't make enough NADPH to regenerate its glutathione shield.
You got it.
So it loses its ability to fight off oxidative stress damage.
That's the chain reaction they uncovered. Oxidative stress then builds up, which is toxic to neurons.
Wow. Okay, so it's like the emergency supplies are locked up precisely when the emergency hits.
That's a good way to put it.
But the study didn't just stop at identifying the problem, did it? They looked for solutions.
Thankfully, yes. They explored what happens if you can overcome that blockage. They focused on an enzyme called glycogen phosphorylase, or GlyP.
GlyP, what does that do?
GlyP is the enzyme that normally breaks down glycogen. So the researchers found that if they could restore GlyP activity, essentially bypassing the tau trap, the neurons were then able to clear that oxidative stress. This led to less toxicity and, importantly, better cell survival in their models.
So restoring GLP lets the PPP pathway work again.
Exactly. It re-enables the production of NADPH, boosts those antioxidant defenses, and protects the neuron. It really shows how crucial it is for neurons to be able to flexibly access these different sugar pathways when they're under threat.
This is fascinating stuff. So bringing it back to the listener, why should you really care about glycogen and tau tangles deep inside brain cells? How does this connect?
Well, if we look at the bigger picture, oxidative stress isn't just a problem in tauopathies. It's a major player in, many neurodegenerative diseases, and even in normal aging.
Right, it's a common theme.
It really is. So this research adds strong evidence to this growing idea that metabolic health and brain health are just, incredibly tightly linked. It opens up potentially a whole new angle for thinking about neuroprotective strategies, targeting how brain cells manage their energy.
Okay. But science is never quite that simple, is it? There are always nuances, caveats.
Absolutely. And it's crucial we discuss those. We need to be cautious and understand the context.
So what are the important buts here?
Well, for instance, you might see discussions floating around about certain molecules, maybe like one called 8-BRCA-MAMP. Some speculate it could activate GLP-1 and maybe mimic calorie restriction benefits.
Okay.
But importantly, that molecule wasn't cited in this specific Buck Institute paper we're discussing. That link is purely speculative at this point. It needs direct evidence.
So be careful connecting those dots too quickly.
Definitely. And similarly, there's buzz around GLP-1 receptor agonists. Those are common diabetes drugs. Some people wonder if they improve cognition by affecting sugar metabolism in the brain.
Plausible, maybe.
Plausible, but again, there's currently no clear evidence they work by changing GLP activity. Claiming that connection now is just hypothetical. We need more research.
Got it. No jumping to conclusions on those drug links yet.
And maybe the most critical nuance, the one that really highlights the complexity, is that the effect seems to depend heavily on the context.
What do you mean?
There was another study back in 2023 in mice. In that study, they looked at healthy but aged mice. And they found that inhibiting GlyP doing the opposite of what helped in the tauopathy models actually seemed to improve cognitive performance in those healthy aged mice.
Whoa. Okay. So inhibiting the breakdown helped in healthy aging, but activating it helped in the disease model.
Exactly. It's seemingly contradictory, but it underscores a vital point.
Which is?
That brain sugar metabolism needs to be, exquisitely finely tuned. What's beneficial might depend entirely on whether the brain is healthy, aging normally, or battling a specific disease like a tauopathy.
So this screams personalized medicine, doesn't it?
It really does. It highlights this crucial need for personalized therapeutic approaches. A therapy that helps a diseased brain, like boosting GlyP in tauopathy, could potentially be unhelpful or maybe even harmful in a healthy brain, or vice versa. Timing and context are everything.
That's a really important takeaway. But despite these complexities, the Buck Institute study itself, the one we're focused on, sounds like solid science.
Oh, absolutely. It was rigorously done. Using that dual model approach the flies, and the human-derived neurons gives the findings a lot of strength.
And they had a clear mechanism, right? Linking the glycogen trapping to the PPP pathway and oxidative stress.
Yes, a very strong mechanistic explanation. And its relevance to actual tau pathology, the kind seen in human diseases, is pretty clear. It definitely, opens doors for thinking about stage-specific treatments, targeting metabolism differently depending on how the disease is progressing. It's a really valuable piece of that huge, complex puzzle of brain health.
Okay, so summing this all up, the big takeaway seems to be that the brain's ability to manage its energy, its metabolic flexibility, is maybe more profound, more important than we realize. And these glycogen reserves might be this sort of hidden defense mechanism, an internal resilience system.
Yeah, a backup plan.
That unfortunately fails in diseases like Alzheimer's when tau gets in the way and locks it up.
Exactly. And the key insight for you, the listener, is that figuring out how to unlock this process, how to restore that metabolic flexibility safely, could genuinely be the basis for a whole new class of neuroprotective strategies.
But with that huge caveat we discussed.
With the huge caveat that any intervention messing with sugar metabolism in the brain needs to be incredibly context-specific, timing-sensitive, and ultimately personalized. It's not going to be a one-size-fits-all solution.
It really makes you think. Given how dynamic the brain is with its energy, what other, seemingly simple metabolic pathways might hold keys to understanding or even treating these really complex neurological conditions?
A great question.
What other hidden defenses are just waiting to be discovered inside us? It just underscores the brain's incredible adaptability, doesn't it? And, the exciting journey of scientific discovery that's still unfolding.